So we're talking about all these different, at this point, covalent modifications, right? We just got done talking about, last time talking about glycosylation, and now lipid modification. And so in this image, the green is representing the amino acid part of the protein, right? And the pink is representing a lipid that's been added on to that protein. So lipids can be added via addition of a covalent bond to an amino acid side chain. When you add a lipid to a protein, a lot of times what happens is it allows that protein to get embedded into membranes, right? Whether that's the plasma membrane or the membrane of an intracellular organelle. And a lot of proteins are embedded into membranes. They don't necessarily span the membrane, they can just have a lipid anchor that attaches them to membranes. So the addition of lipids, again, there's enzymes that add these lipids, and that process often occurs in the endoplasmic reticulum. So the take home, lipid modification refers to covalent addition of a lipid to a protein, and it oftentimes allows them to have a tight association with the membrane. Okay. So there are other modifications, but for this class, for this lecture, we just focused on phosphorylation, glycosylation, lipid modification, kind of moving on. And the rest of the semester, we might come across other types of modification, and I'll point it out when we get to it and say, hey, check it out, it's another covalent modification. And when you think covalent modification, what do you think of in general? What does that mean? What is a covalent modification on a protein? It is, so first, okay.
Is something that's reversible?
They are almost always reversible, except for, starts with P, proteolysis. Proteolysis is actually considered a covalent modification because you're breaking a covalent bond between peptides. Yeah, and so it's unique because it's not. It's irreversible. Every other covalent modification is reversible. How does it get reversed? How do they get added? What's doing it? It just manifests itself or enzymes. Boom. Covalent modifications means the addition of some chemical group, unless you're proteolysis in which you're just creating something, but typically addition of some chemical group, could be lipid, could be phosphate, could be a whole big old sugar, polymer, oligosaccharide. Added by an enzyme, removed by an enzyme, right? It's the only way these covalent modifications occur. Addition with the help of an enzyme, removal, again, with the help of an enzyme. So you need an enzyme to help create the covalent bond or break it. All right, so now a lot of proteins their function can be regulated by the covalent modification that occurs, right? So you can have something simple, like in the slide where I introduced kinases. So maybe a protein is active once it's phosphorylated and then inactive when it's dephosphorylated, right? It can also be much more complex than that. So here's a look at a PowerPoint version of a protein called P53. So P53 is a really important tumor suppressor gene. So it's involved in a lot of cancers. It's disrupted in a lot of cancers. The function as a transcription factor. It has cytosolic roles as well. There's a lot of different potential jobs in the cell. And the way it performs any one job is determined by how it's covalently modified. Okay, and so on this diagram, what they're showing is these different domains, and over here there's this domain where it has all these potential sites where it can be phosphorylated, right? 1, 2, 3, 4, 5, 6, 7, 8, whatever. A bunch of sites where it can be phosphorylated, right? Down here, there's a bunch of other sites where it can be covalently modified, but in different ways. So there's a key here, right? So P53 can be acetylated,
Phosphorylated at these two sides. It has some other funky covalent modifications, so it can be monoubiquitolated or sumoylated. So I'm not going into the details of these specific types of modifications that we haven't talked about yet. The point of this slide is to talk to you about how P53 can have different functions. The way it performs those functions is dictated by what are the combination of modifications that have occurred on it, right? So this is referred to as having a combinatorial code, right? The code is read and then it performs that function, right? So here it shows 4 kind of generic functions of P53. So binds to proteins Y and Z. Maybe in order to bind to proteins Y and Z, this whole region needs to be phosphorylated and nothing can happen down there, right? So that means that P53 would bind to proteins Y and Z. Maybe no phosphorylation, but acetylated only, that causes it to translocate to the nucleus. It can go then perform all its nuclear functions. Move to proteasome for degradation. My guess is going to be there's polyubiquitylation that can occur here to cause that to happen. Or move to plasma membrane. Maybe that's what sumoylation is. I don't know. I'm making up. I don't know all the details of P53. That's not the point, right? The point is that there are all these potential modifications, and there's a specific subset of them that will cause P53 to move to the plasma membrane. A specific subset that will cause it to move to the nucleus or to bind to different proteins. That's how it gets to have so many versatile functions. Because anytime you have a different subset of covalent modifications, what's being affected differently? The protein structure, right? If you just phosphorylate it, that's going to cause the protein structure to shift in some way. And then if you add some acetyl groups too, it's going to cause it to shift in another way. If you add a sumoylation, it shifts in another way, right? And every time it shifts, it can have a different function. Or a sweet robot dance. Yeah, no. So the set of any one protein, so this example is P53, a lot of proteins function this way. The set of covalent modifications that occur at any given time can dictate a different functional outcome, right? So combinatorial regulatory code, right? That's a fancy way of saying the different combination of modifications can regulate the protein's function. So different combinations of modifications on the same exact protein equals different functions. It's gonna cause it to have slightly different structure. So because I think me talking at you about this, so some of you are like, I don't know, and others are like, no, I get it. I did this little activity just for you guys to do on your own. And this is just a very stripped down example. So instead of giving you like 40 different potential modifications, there's 3. Sometimes that's the case. Sometimes a protein just has 3. And in my made up example, we have this protein NGN in orange. It has 2 potential phosphorylation sites, right? It's site 1 and site 2, and 1 potential acetylation site. And so this is NGN's combinatorial regulatory code, right? I gave you the key. When the protein NGN is phosphorylated at both sites, it translocates to the nucleus. When it's phosphorylated at position 1 only, it's degraded. When it's only acetylated, it binds to another protein in the cytosol, right? So I'm just requesting that you, on your own time, just draw that out and think about what that means, right? And you can even draw NGN having a slightly different, like, wiggly structure because of the specific combination of modifications. Okay, we can do number 1 together. When the protein NGN is phosphorylated at both sides, it translocates to the nucleus. So what I want you to draw here. Yeah. I just want you to draw NGN being phosphorylated here and here, not acetylated, and you can perhaps draw it in the nucleus because of that, right? Get as detailed as you want, glitter pens if you want, I don't know what you like to draw. But yeah, just draw it out, because a lot of people, they come to me, they think they get this, and then they come to me before the test, and they're like, oh, regulatory, what's happening? So I created this simple activity to try and help you just draw it. And you may be going, this is the dumbest thing ever, but trust me, it will help you understand. Okay, so next, proteolysis, right? We talked about this example last time with insulin. It is, and there are a lot of proteins like this, that when they're first translated, they're not active yet. They need to be proteolytically cleaved in some specific way in order to become active. So, right, insulin, its first, in its initial state is called proinsulin. Passive form leaves very specific disulfide bonds. That allows it to be cleaved at these specific sites. And in this case, proteolysis is activating this protein. We're going to talk about other examples of proteins that also require to be proteolytically cleaved to be active. And as you learned when I was talking about protein folding, right, proteolytic cleavage can absolutely also stop a protein's function as well. So proteolysis refers to the cleavage of a protein, and it can be required to both start a protein's activity or to stop it. So in this way, right, if you inhibited the ability of proinsulin to be proteolytically cleaved, you wouldn't have any functional insulin, right? When we learn about how apoptosis is regulated, so that's regulated by a group of enzymes called caspases. They're initially formed as procaspases. And the way the cell regulates whether or not apoptosis is triggered or not is you either facilitate proteolytic cleavage of those caspases or you prevent it. Good. Okay. Allostery. So allostery you may have heard about before in reference to enzymes. Those are the proteins that this allostery was first identified in. And at its very core, it's basically a change in the protein at one site that's influenced by interaction with something at a different site, right? There are two sites on the protein that are involved, right? So on an exam, like on the practice exam, right, there's something about, on the practice exam that's posted, there's a question that it's embedded in the answer, like which of the following describe allostery? And the two answers in there that are correct, they both involve influencing binding of a substrate at one site by influencing or by association at a different site of a different protein. It takes a lot of words to say it, which is annoying because what's happening is very straightforward. Binding at one site either induces or prevents binding at another site. There's a bunch of slides dedicated to this, so I'm going to keep going. So again, this is first identified in enzymes. And that's what's kind of shown here, where you have an enzyme that interacts with a particular substrate. And this was first identified when they noticed that there was what's referred to as an allosteric regulator or effector that bound to a totally different site on the protein. And sometimes what you see happen is negative allostery, where when that allosteric regulator binds, it inhibits the ability of the enzyme to associate with its substrate. Right? It changes the conformation so that that substrate gets kicked out of the protein. It can also happen where you have something called positive allostery, where binding of an allosteric regulator changes the conformation and invites the substrate in. There's two things that can happen, but it always means so the allosteric protein is the one that is going to be influenced by an allosteric regulator and some other protein that it's interacting with. And it has two distinct sites in which the allosteric regulator and its target substrate associate with. So allostery in the field of molecular biology and cellular biology is kind of expanded, and it's thought to apply to most proteins. So a lot of proteins, you see this phenomenon where some other molecule or protein interacts with the protein at one side, and it affects its ability to interact with another protein at a different site. The key point, again, it always involves two different binding sites on a protein being regulated. Now we're going to walk through an example of both positive and negative allostery. So positive means that the allosteric regulator is positively influencing binding of that other molecule. Okay. And again, we're talking two distinct binding sites. And I have two children who have a strong affinity for dinosaurs, so I really appreciate that this looks like a T-Rex head. Right? I know. So in this case, we have molecule X is functioning as like the allosteric regulator, and then this protein and its other binding site associates with glucose. So in the absence of molecule X, we have the open T-Rex conformation, which doesn't really favorably bind glucose. So you see some binding happening, but not very much. So in an experiment, if you were to then add molecule X, or in the body, if all of a sudden molecule X was present, it binds to the T-Rex I, which is distinct from the T-Rex valve, where glucose binds, like two distinct sites. Right? You get the molecule X binding to this distinct site, and it shifts the conformation of the protein. When it shifts the conformation of the protein, it changes it to a conformation that really favorably binds glucose. So now you see increased binding of glucose. So molecule X helps influence positive binding of glucose. Okay. And typically, when you see these kinds of positive allosteric interactions, really both molecules can help drive binding of the other one. And so sometimes this is also referred to as cooperative binding. So I know that I think in Bio 2 they introduced hemoglobin's binding of oxygen as cooperative binding. That's a type of positive allostery. And we'll talk about that example after I describe negative allostery, which is just the opposite, right? In negative allostery, you see a negative coupling between these two. So in this case, sticking with the same T-Rex space, the glucose binds just fine on its own. So the protein itself naturally has a conformation that can bind to glucose really easily, unless you have molecule X present. In that case, molecule X changes the conformation so that the protein releases glucose. Okay. So in one case, you have an allosteric effector increasing binding. In the other, you have an allosteric effector decreasing binding. And in both cases, it involves distinct sites on the protein. Yeah?
Will there ever be a situation where they're both present?
Yeah, and in that case, guess which one would win? There's two things that
Dinosaur.
I like it. So what happens really is if you have two proteins that want to associate with T-Rex face, the one that wins is usually the one that's in a higher concentration or has a much higher binding affinity. Yeah.
For allostery, is it only two sites or two or more?
Yeah, there can be more than two. Yeah. I just don't get it. I think when I get to GPCRs, I'll talk about how to get to GPCRs. But yeah, there can be more than two sites. For this class, I'm going to talk about the two sites. Yeah. Yeah, someone asked over the weekend about multicast transmembrane proteins. These proteins, G protein coupled receptors that wind back and forth across the membrane 7 times. They're huge, and they can interact with a ton of different proteins, and they have all this rich allostery that happens, kind of traverses the membrane, right? So things binding on the outside can influence binding on the inside. Yeah. Okay. So hemoglobin and oxygen are a really famous example of cooperative binding. It's also an example of positive allostery. And so hemoglobin, right, it's tetrameric. You have the 2 beta subunits and the 2 alpha subunits, and every single subunit has a heme group, right, this blue disc that binds oxygen. And the way the kinetics of oxygen binding are amazing, and I don't remember the exact numbers, but the first oxygen that binds is the slowest to bind, and it shifts the conformation so that binding at the next subunit is faster. That shifts the conformation so at the 3rd subunit it's even faster. And so by the time you get to the 4th subunit, it's like 400 times faster or something crazy like that. So it's this cool mechanism that where basically the effector is also the substrate that's binding, right? So, and that can happen, right? Don't get too hung up on semantics of allosteric effector and substrate. It's really just the 2 distinct sites. Oxygen binds, shifts conformation, which increases binding of the next oxygen. That shifts the conformation again, increases binding of oxygen to the next one, and so on. Yeah?
Does it change the conformation of a whole molecule or just the site mix?
Yeah, that's a good question. I think it's shifting the whole molecule every time. And so this is part of the mechanism of how hemoglobin picks up oxygen really quickly in areas where the oxygen concentration is high, which would be right in the lungs, where you need hemoglobin to pick up oxygen super fast. And why, when it gets out to deoxygenated tissues, has an easier time releasing that oxygen. CO2 comes into play too. That's probably more a systemic physiology topic, but this is part of what enables hemoglobin to suck up oxygen really efficiently in the part of the body where oxygen is highest. Okay, you guys get allostery, positive versus negative? 2 different sites. If I don't say something about different sites on the test, it's not allostery. Okay. So another way that cells, a way that cells can control the activity of an allosteric protein is to control that allosteric effector, the regulator, right? And so I'm going back again to this example of the sarcomere. And I'm going to copy this image on the board because I'm going to come back to it again. So let's walk you through what the heck we're looking at first. Here is the axon terminal, right? So here's where acetylcholine, which is the neurotransmitter that's super important for triggering muscle contraction, here's where it's released, right? And that is received by the muscle cell, and it actually triggers an action potential that culminates in causing calcium to get released by this structure called the sarcoplasmic reticulum. Sarcoplasmic reticulum is basically a fancy ER in muscle cells. And it sequesters, it's famous for sequestering calcium. And so I need 2 colors here. I only need 3 colors, maybe 4. Let's just see what happens over here. So I'm going to draw that sarcomere. I'm just going to draw part of it. So we have our myosin filament, and then we have actin forms helix of a bunch of globular proteins. here. The myosin heads. Right, again, these heads need to walk along the actin. Actin filament. This is part of a sarcomere. Okay, we're gonna get to the molecular mechanism of how this is triggered. So, what you also see is binding of this protein called tropomyosin. Around the filament, right? You guys can follow the image on your slide more than the image that I'm drawing up there because it looks better, but I'm doing the best I can. And these other proteins on it. Okay. So ultimately what we want is for myosin to walk along the actin filament. That's what facilitates contraction. And what I'm going to tell you right now is we need calcium in order for that to happen. So basically, the muscle cell sequesters calcium in the sarcoplasmic reticulum, and when it's released, calcium to enable association. Oh my gosh, I'm sorry, this was supposed to look nicer than this. Between actin and myosin. So calcium is actually an allosteric regulator of troponin. So calcium allosterically regulates troponin. So what's happening here, myosin wants to walk along the actin filament, right? That green braided protein. Tropomyosin. Blocks myo absence of calcium. Right? So when calcium isn't present, tropomyosin, right, is directly blocking that myosin from binding to actin. So it can't walk along it. When calcium is present, it binds troponin, and troponin has a conformational change that causes it to move tropomyosin out of the way. This is bless you. people have had systemic phys already. Okay, so for those of you who are like, wah wah, review. But, right, so that is the beautiful molecular mechanism of how contraction is facilitated. So, right, you have calcium being hidden in the sarcoplasmic reticulum until you want, which means that troponin is just holding tropomyosin in a conformation that just blocks the binding site of myosin to the actin, so it can't walk along it. When a neuron stimulates contraction, what it's really doing is stimulating the release of calcium from the sarcoplasmic reticulum. Calcium then interacts with troponin, which really is causing a conformational change in troponin that causes it to shift tropomyosin out of the way. It changes, right, its association with tropomyosin at a different site on the troponin complex. It's actually a protein complex. That make sense? The heart of this is how the cell regulates the activity of the sarcomere by either hiding or releasing this allosteric regulator. I'll leave that up for people who are hard to see. Okay. Yeah.
So is there a binding site on the troponin?
Yeah, so troponin, basically, it's a complex, and at one part of it can bind calcium, and the other part of it, it's binding tropomyosin. So when, in the absence of calcium, the way troponin is holding on to tropomyosin, it's just holding it right over that myosin binding site. But then when calcium binds over here, it goes, and it shifts the conformation of the protein. It shifts the way it's holding on to tropomyosin. It basically just pushes it out of the way. And cool, now myosin can bind and walk along actin and cause contraction. Okay. So moving on from allostery, another way a cell can regulate protein activity is it can provide or prevent a binding partner that's necessary for activity. So one example that we're going to go into more detail on when we get to signal transduction facilitated by something called receptor tyrosine kinases. These are receptors that sit in the membrane, and they're important for signal transduction, right? So a ligand needs to bind, and then it transduces the signal to the inside of the cell. Well, the ligand, the way it affects activity is it's something called enforced proximity. When it binds, it actually causes those receptors to dimerize. So it brings them together. So the take home here is if the cell doesn't want these proteins to be active, no signaling molecule, right? When you want the signal transduction pathway to be active, you get production of that signaling molecule, and it will bind to the receptors and enforce dimerization. And that leads to a mechanism that, right, this is the big picture that I want you to understand. The actual mechanism of what's happening with the phosphorylation, we'll get into more detail when we get to signaling. The way in this case that enforced dimerization activates these proteins is they cross phosphorylate one another. They have enzymatic activity. It only happens when they're brought together. Yeah.
So it's like non-allosteric because the tyrosine kinase domain acts as one binding site together.
Yeah, in this case, it's what the signaling molecule is doing is actually smushing them together, and it's just the sheer proximity of them being close to one another that causes them to be active. So instead of the binding causing a discrete conformational change, it's just bringing them together. And there are all sorts of cool experiments that have been done to look at this, where they've just created like a mutation or kind of a linker in the proteins that just forces them to be together all the time, and they're just all active. So it's not necessarily some specific structural change that's happening because of the binding
However, there's probably some gray area in a lot of these examples, okay, that I don't go into because it just gets a little bit too muddy for, a little bit too muddy for here. But fun office hour conversation. All right. And then here's a subject that gets a little bit confusing. So cells can also, right, affect a binding site. Affect a critical binding site required for protein activity. So now we're getting back to the whole troponin, tropomyosin situation. So if you were just looking at what tropomyosin is doing, tropomyosin is not really, is not regarded as the protein that's being allosterically modified here. It's troponin, that is. So tropomyosin itself, what it does is it just blocks the binding site of myosin to actin. So that is specifically regarded as blocking a binding site. And that happens all the time. When you have a protein that you want to interfere with its ability to bind some target protein, you interfere with directly interfere with the binding site. So instead of allostery, where you have a protein with you're talking about two distinct domains. Now you're talking about one. Okay. Right, there's two sites involved here, and in this case, the way the cell regulates actin-myosin, so here's myosin binding site. I know, why does it, so actin is really a bunch of globular subunits, in case, because a lot of times when I draw this, people were like, but I thought you were drawing DNA. This is actin forms these long filamentous polymers. So myosin binding site, and what's happening is you have troponin blocking that one site. So when you have a situation where you have myosin, did someone just say what? What are you doing up there? Myosin is a motor protein. I've always talked about being a motor protein for how long we don't have this time. I might look like something else though. I'm just saying, that's all good with me. I don't want to be weird. Yeah, well, that kind of thing. Yeah.
Is it troponin that blocks myosin?
Tropomyosin. Tropomyosin is what physically blocks this binding site, okay? So here, I'll get there. So myosin, and did I just, that's why you said that. Thank you so much.
Yes, there you go.
Oh my gosh, no,
There.
that's what the what was. It wasn't my ugly
Tropomyosin.
protein, it was,
There you go. I was like,
oh my
yes.
gosh, thank you guys. Myosin. Everyone in the room knows I just made that mistake, okay? Okay, yeah, okay, so tropomyosin is blocking. So myosin, and we'll get there. One of these, I was just saying off hours, one of these days I'll stop making mistakes. Probably when I'm dead. Hey, morbid but true, right? Like life is making mistakes and doing what you can to improve. Competing for the same binding site. Yeah, I already referred to death and looking like Miss. Doing good up here. All right, so does that make sense? This involves one binding site. Tropomyosin blocking this binding site that myosin needs to interact with. It just so happens that in the whole mechanism, you also have, when you're talking about the moving of tropomyosin, an allostery involved, right? So there's a bunch of things happening in the context of a sarcomere. It's complicated, right? We're all very smart people, and I think together we can get there. Okay, so this part, blocking a binding site. Okay, when you talk about that being moved, now you're talking about an important allosteric mechanism in which you have troponin, one site. Okay. So one site binds tropomyosin, the other site binds calcium. When calcium binds, it moves tropomyosin out of the way so that myosin can bind. So does that make sense that in this case we're talking about blocking of a direct binding site? That's not the part that is allosteric. It's just directly blocking myosin from binding. The allosteric part comes in with how troponin moves tropomyosin out of the way. Because troponin is bound to tropomyosin at one site and it interacts with calcium at a different site. When that happens, it shifts tropomyosin out of the way. Does that make sense? Okay. Yes, I can, absolutely. I'll get out of the way.
So lost.
All right. Okie dokie, right? And this question, right? So what causes the, and I saw the word release, like when I was reviewing my slides before class, I don't like the word release because it's not that tropomyosin totally releases actin, it displaces it, or it's displaced. It's just shifted out of the way, if that makes sense. So I just changed the word, I left a slash there so no one freaked out, but really it's displacing tropomyosin. And what is it that's displacing it again? Troponin, and it's when troponin specifically is allosterically regulated by, got it. So another thing a cell can do to influence a protein's activity is affect the polymerization of a protein that needs to function in the context of polymer. So actin is one of those examples. So I do, it's a bunch of globular proteins that form this heel pull arrangement. So actin always functions in the context of a polymer. Here, those actin filaments, also with myosin, even though they're not drawn here, form the contractile ring, right? That separates two cells at the end of division. So in order for that contractile ring to function, actin needs to be able to form those polymers, as does myosin, to create the appropriate ring structure that then constricts in and allows those two cells to separate. So, right, in any situation where you have a protein like actin that needs to function in the context of a polymer, if you prevent its ability to polymerize, you're going to destroy its function. And in the cytoskeleton lecture, which is after exam 2, we're going to talk about all these proteins that are required to build up actin and break it down. And how it's really all about those proteins manipulating actin that give actin its particular jobs.
Good. So one little actin globular subunit on its own, worthless. If you don't want actin to perform a job, cause it to depolymerize. In fact, that's what happens at the end of division when the contractile ring is done separating the two cells. You don't need that contractile ring function anymore, right? So the cell induces the depolymerization of those actin filaments, break them up into its subunits so that those actin subunits can be recycled to do something else. Okay, last but not least, the protein structure and function affecting scaffold complex components. Really, this slide is to help introduce the idea of a scaffold complex. Has anyone here heard of a scaffold complex before? I have my Charles and T's for you. You guys want to
I have.
Okay, I have two. Okay, so a scaffold complex is a group of proteins that are designed to work together. Right? So they're useless without one another, right? It's like you complete me. It's true, they complete each other. Useless on their own. And usually at the heart of a scaffold complex is one big old protein that's regarded as the scaffold protein that's kind of helping to hold all the components together. So this right here, one of the examples is the ubiquitin ligase complex. So we talked about the importance of polyubiquitination in targeting proteins for degradation. So it's this complex that actually does that. So here's the way that protein crystallographers like to present protein structure, kind of showing this organized scribble of proteins that kind of show where the alpha helices are, beta pleated sheets. Here's a more straightforward PowerPoint version of the scribbles to help convey what's happening here with ubiquitin ligase. So you have the scaffold complex, right? It itself is just kind of functioning to hold everything together in a particular conformation. It also has two adapter proteins, same thing, kind of you have this big old scaffold, and in the case of ubiquitin ligase, it has two adapter proteins that adapt the association of the actual ubiquitin conjugating enzyme. So this is going to be the enzyme that actually adds each of the ubiquitin subunits onto the target substrate. And then it has the substrate binding protein that, say it's binding a misfolded protein, right, and targeting it for degradation. It's going to be held in place right here. So target protein in brown would be held on by the protein adapter, and then the ubiquitin would just get added as it holds it in place. And so if you think about it, if you lose any one of these, you're not going to get ubiquitin activity. You lose the scaffold, well, these don't know how to associate in the proper conformation. If you lose the red adapter, you lose the ubiquitinating enzyme. You lose this adapter, you have the ubiquitinating enzyme, but you don't have the arm that's holding the substrate in place. So the take home is if you have a scaffold complex, you need every single one of those associating just so to get to complex activity. So, I have that all already. So, if any protein is prevented from associating with complex, it can't do its job it's designed to do. How could you prevent association? What are some of the things you might do to one of those proteins to mess up its ability to associate with the complex? Ooh. Okay, how about raise hands? Because lots of people have great ideas.
We're attaching onto the ligand, or actually the ligand to the.
So you're talking like a mutation, whatever. Yeah, for sure. Mutation, change the sequence, change the structure, for sure. What's another way outside of a mutation? So you already have these proteins made. What could you do? Oh, I think we got.
You could modify the structure.
so change the conformation of any of the parts such as
Yeah, and what's some of the ways that you could change the structure?
phosphorylation.
Boom, phosphorylation. Did you have another one?
Lipid.
Lipid modification, yes.
modifications,
Any covalent modification, right? Any covalent modification to any one of these might affect its ability to associate with the other scaffold components. Yeah, or a mutation in one of them, or you prevent expression of one of them. Any manipulation to any protein in the complex might affect its functional output. It's a take home. And then that same exact message is on the 2 subsequent slides. So we're gonna talk about, so the take home, right, there's a big old scaffold, just like here in green. So the whole thing is a scaffold complex, and in a scaffold complex, usually one of the proteins is called the scaffold protein. It is kind of the big daddy or mommy, I don't know, whatever, that's holding everything together. I don't know, weird analogies. So in this case, you see scaffold complexes a lot in the context of signal transduction. You have a scaffold complex that's functioning to hold intercellular signaling molecules right next to a receptor. So this whole thing would be regarded as a scaffold complex. If you lose any one of them, it's not going to function in the same way, right? Because scaffold proteins holding it all in position, and then the other components, if you lose one, well, it's necessary for transducing the signal from the receptor to 2. So if you lose one, 2 is useless. Same thing if you lose 2, you might get 1 activated, but you don't get 3, right? So you need all the pieces of a scaffold to work. And then finally, I just felt like putting in my favorite example of a scaffold. I'm not testing you on the details of the synaptic scaffold, but it's a synaptic scaffold. Guess where you find the scaffold complex? Yeah, and synapses. So this is at the postsynaptic membrane. So you find this in dendrites on the receiving end of neurotransmitters. And you have this big old scaffold protein that helps hold all these scaffold components in place. If you lose any one of those, it affects the ability of the dendrite to receive neurotransmitter signals appropriately. In fact, neuroligin, that's one of the common mutations that's found in autism. Okay. Membranes. You guys know what organelle that is? Mitochondria. Yeah, why'd I use that one for the image for biological membranes? That's 2 membranes. That's a lot of membrane going on. Review what we're going to talk about. Talk about membranes, what they're composed of, what are their functions, what are their hopes, what are their dreams. Very tired. Okay. 20 minutes? No. 19 minutes. Do this. Right, jumping jacks. Caffeine IV. I was talking to one of the, Dr. Wright, who in his systemic phys class, he passes out chocolate covered espresso beans. Yeah, he's like, well, yeah, I just go to Winco at like 4 in the morning and buy them. I'm like, what? No, okay. You already lost me. Like the chocolate covered espresso beans, you had me. The 4 a.m. Winco trip. But yeah, I feel like, I don't know, I'm torn on whether that's a good or a bad idea. It'd be a bad idea for me. I'd be like, no, okay. So all cells, whether you're a prokaryotic cell or a eukaryotic cell, they all have a plasma membrane, right? Prokaryotic cells only have a plasma membrane, right? They don't have membrane-bound organelles. So prokaryotes can have cool features that allow, they've identified like these protein lattices that can kind of sequester components into one region of the cell. But so far, we haven't identified any membrane-bound organelles inside of prokaryotic cells. Eukaryotic cells, on the other side, have maximized use of their membrane. So all organelles are membrane-bound. And when people talk about this, they always refer to the fact that this allows our functions to be compartmentalized. It allows for increased kind of refinement and complexity of those functions. So to think about that, if that doesn't outwardly make sense to you, think about, right, a cell or any of these organelles, they're kind of like little factories, right? And each organelle has its own function, right? So think about something else that's made in a factory, like something we can all relate to, like a car. Does anyone know how cars are made in a factory? Do you have like an entire car that is made in any one factory? No. What actually happens in reality? Does anyone know?
Different parts.
You have parts. You have different parts made in different factories. Why do you think that is? Allows them to specialize in just that part and be badasses at making just that part, right? You can specialize, and so you know what you're doing. If you had a whole car being made in one factory, it'd be chaos, right? So it's the same kind of phenomenon in a cell. It allows you to make sure that all the enzymes that are specific to performing refinement of glycosylation, for example, the refinement of glycosylation really occurs in the Golgi. So you can house them there, so they don't have to they're not getting in the way of, or other things aren't getting in the way of them doing their job. That's how I think of it. I know a lot more about how cells work than car factories, but unfortunately, okay. So what do we need membranes for? They are important for a lot of things. So, right, we just talked about compartmentalization of cell functions. It allows different functions to be compartmentalized within the context of a specific domain of the cell. The membranes provide defense and integrity of the cell or compartment. So cell, I'm referring to the plasma membrane, right? It's going to be the one protecting the whole big old cell. Compartment, I would mean like individual organelles. So the membranes, right, are semi-permeable barriers, right, that only allow passage to certain things. It's very organized and regulated. Selective permeability, regulation of internal cellular compartment activities, right? So when I talk about the membrane, I'm not just talking about the lipid bilayer, I'm talking about all the proteins that are embedded in that membrane as well. And so all those proteins perform really critical functions that can dictate cellular activity. When cells attach to the ECM, or when you have so a cell move, a cell migrate, or an organelle in the cell needs to move, it is always facilitated through the membrane. So for the example, in cell migration, you have receptors that actually bind to regions of the extracellular matrix, and it's that binding and release of the extracellular matrix that allows the cell to attach and move along the extracellular matrix. So it connects via proteins in its membrane. So the membrane, therefore, is critical for attachment and movement of cells and compartments, and responds to signals from the outside of the cell or compartment, right? You have all these receptor proteins that sit in the membranes that can receive signals and have a response according to what they bind. All right, so I just kind of mentioned this, the main components of biological membrane. It's not just the lipid bilayer, though that is a critical component of it. So the lipid bilayer is referred to as amphipathic because it has both hydrophilic and hydrophobic components. It also has membrane proteins. A lot of those proteins have big oligosaccharides that perform important functions as well. Cholesterol is a really critical component of our membranes that helps provide rigidity to those membranes. And then the submembrane protein meshwork. So here's an image of a membrane, right? So here's the cytosol, here's our membrane, lipid bilayer, big green proteins stuck in the lipid bilayer. There are also a bunch of peripheral proteins that are connected to the membrane, but not spanning the membrane, right? Like remember I talked about how lipid modified proteins can be kind of anchored by their lipid attachment into the membrane. There's a ton of those. There's also a rich cytoskeletal network just underneath the membrane, super important for the structure of the membrane. That also counts as the cytosolic submembrane protein measurement. I didn't say it slow for emphasis, I said it slow because it's 4:03 and that's a lot of big words. I'm sleeping. Nap. Whenever my kids fight naps, I'm like, how are you possibly fighting this? Do you know how much mom would love to be forced to take a nap every day? Oh, kids. They also fight food all the time. I'm like, but you need this to live. I don't understand. Like, how do I have to? Okay. All right, so we're gonna take a closer look at each of those components. Hopefully a lot of this is reviewy, it's not too dense for the final 10 minutes of lecture. So amphipathic lipid bilayer, right? So we have phospholipids, and phospholipids are named that way because they have a hydrophilic, polar, phosphate-based head. We'll look at that on the next slide or in a couple slides. And then hydrophobic fatty acid tails. And so the lipid bilayer provides this hydrophobic barrier to most molecules. So small hydrophobic molecules can pass pretty easily. A lot of nonpolar gases like oxygen, CO2, nitric oxide, those things can pass through pretty easily. Water can pass through, urea. But most things can't just pass through the membrane, right? It's regulated by proteins that either provide or prevent passage. So this slide is just showing the kinetics of molecules moving across a lipid bilayer. So again, in this case, no proteins present. We're just talking about if you just had that lipid bilayer, how fast would these different classes of proteins get across? So again, hydrophobic nonpolar molecules can cross that lipid bilayer super easily, right? Hence the 5th green arrow. These guys are like, woohoo, we're in. Small uncharged polar molecules, so like water, it's not charged, but it is polar. Urea, glycerol, they're pretty small, so they can still make it through, though they not as easily as the hydrophobic molecules. If you're a large uncharged polar molecule, this slide, right, so technically can cross even more slowly, so you left them in there for a super long time, you might start to see some on the other side of the lipid bilayer. In reality, in us, these molecules rely on facilitated diffusion to cross a membrane. Okay, and then if you're an ion, you're just, let's go back to that acronym you guys learned about earlier in the semester, your SOL. You're poop out of luck. Should I do POL to make it more figurative? Okay, just keep it SOL. Makes me edgy. I am not edgy. Right. So cool. Oh my gosh, okay. So, charged or strongly polar molecules like ions and sugars. In the case of proteins, right, there are some proteins that aren't necessarily charged, but they're just so big, they can't just move across a lipid bilayer. And so this really, the preventative passage of ions is what allows us to have what are called membrane potentials, right? It allows us to have a charge differential on either side of the membrane, which is important in a bunch of different cellular contexts. The one that is my favorite, right, is action potential. So if you have a differential of charge on either side of the membrane, it allows you to use that charge to create kind of energy currency or have potentials down membrane that you see like on muscle cells and hair cells. All right. Here's your friend, the phospholipid. So it's broken down into kind of three parts. I'm going to draw this over here. Right, so the glycerol backbone, so those of you who don't know glycerol, Glycerol is just 3 carbons, right, that have hydroxyl groups off of every carbon, you know, hydrogen everywhere else. Okay, I drew the oxygens this way, or the hydroxyl groups this way, just to be in line with the way you see it here. So the glycerol backbone is right here, these 3 carbons, and then if it's just glycerol, you would have hydroxyl, hydroxyl, hydroxyl. What happens in the context of a phospholipid is a polar phosphate head group is attached right here. So you have polar phosphate head group. That is what when they draw the phospholipid having like a circle at the top, it's encompassing glycerol backbone plus the phosphate head group. And then down here is where you get the fatty acid tails attached. Right? So you have fatty acid tails. That is the super basic structure. Fatty acid tails. Now, those fatty acid tails can be saturated or unsaturated. A lot of times you see one being unsaturated, and when you have the unsaturated, I'm doing this stupid dance because it kicks out and it allows there to be a little bit more space next between it and the next phospholipid. So it creates a little bit more fluidity in the membrane. If you have 2 saturated fatty acids, they like pack in like sardines, right? And so you don't have as much fluidity. And you see both in biological membranes. The most common phospholipids will have a unsaturated fatty acid tail and 1 saturated fatty acid tail. They kind of strike a balance. But again, both are possible, both are safe. The polar phosphate head group. Check out how you have the phosphate and then the R group up here. Oh crap. R group, what does R group usually mean?
Amino.
Yeah, right, so it can vary a lot. And that's at the heart of this slide. No, I don't want you to memorize these structures. What I want you to appreciate is that there are different types of phosphate head groups. Right, so right here we have again the glycerol backbone right here, 2 fatty acids, and here are the polar phosphate head groups. And so I just want you to appreciate that this one is CH2, and H2. This one is different than that, and this one is different from those 2. I want you to appreciate that there are a lot of different phosphate head groups. Because a while back, we thought that all phospholipids in the membranes were the same and they weren't that big of a deal. And it turns out, holy crap, we were super wrong about that. Yes?
So, does it just depend on the type of membrane?
Yeah, you see a lot of variation in membranes. So there are hundreds of phospholipids. And there are kind of big families of phospholipids. So, phosphatidylethanolamine, phosphatidylserine, phosphatidylcholine. These are common ones that you'll see, but there are a bunch of different ones, and you'll see variation within, is it the plasma membrane? Is it the Golgi membrane? Is it the ER? You'll see variation, and you'll see variation in the 2 leaflets of the membrane too. And this is beyond the scope that we're gonna talk about. In like the Golgi, you'll see something. Has anyone ever heard the term lipid raft? Lipid raft is where you'll see like a microdomain of phospholipids. You hear the term microclimate? Like in the Bay Area, you have these microclimates. It'll be like 90 degrees in Palo Alto and then like 60 degrees in Colma and South San Francisco, because the Bay Area has all these microclimates. A lipid or a biological membrane can have microdomains of phospholipids where all of a sudden in one region you'll just see a ton of phosphatidylcholines. That's called a lipid raft. And that is, it's turning out having a super big impact in what's happening in the cell at that particular region. Lipids are really important. And I think as this class moves forward, I'm gonna try to get more lipid biology into the class as the scientific community learns more about them. Oh my gosh, but for so long, it's like proteins do everything, and now we're like, oh crap, no, there's things called microRNAs that also are really important. Oh crap, these phospholipids, they're not just these amphipathic molecules that are serving as a barrier. They're really important for what that membrane is actually interacting with and where it's interacting with. Fatty acid tails can vary too. So fatty acid tails, instead of being, so each of these points represents a carbon. So you can have different lengths of carbon chains on those fatty acids. This is representing a double bond. So that's, sorry, I forgot to mention, an unsaturated fatty acid is one that has at least one double bond. Saturated, all single bonds. So you can see some will have saturated, some will have unsaturated. So don't think about phospholipids as just these monolithic things that are exactly identical. They're not. What's identical is, yeah, they have a polar phosphate head group, they have a glycerol backbone, and they have fatty acid tails. But besides the glycerol backbone being the same, those parts can vary a lot. And so this slide is just trying to get that point. Each of these colors is supposed to represent, sorry, it's hard to see now, the different phospholipids, right? So phosphatidylcholine is red, sphingomyelin brown, phosphatidylserine puke green, right? And so what this is trying to show is that not only do you have different phospholipids in a biological membrane, you can have different ones on different sides of the membrane. We see that all the time. There are actually enzymes in the ER that when phospholipids are produced, their job is to position them on one side of the leaflet or the other. So each of these sides are called leaflets. And so there are these enzymes that have cool names like leafletases and phosphases and scramblases that their whole job is dedicated to positioning these lipids. And we'll pick up here.